So today in the inorganic lab we are going to perform the limit test of iron using sodium chloride or NaCl. So here the principle of this experiment is that iron in Fe2 plus form reacts with thioglycolic acid in presence of citric acid and ammonia solution to produce a complex which is ferrous thioglycolate, and the color of the complex is pale pink to deep reddish purple. The color of this complex is compared between the test and the sample solution and the standard solution. So this is basically the principle of this limit test. So here we will be preparing test and standard sample solution simultaneously. In case of test sample, we will take 1 gram of sodium chloride and dissolve it in distilled water and make up the volume up to 40 ml. Then we will add 2 ml of 20% weight by volume of citric acid to it. After that, 2 drops of thioglycolic acid are added and the entire solution is made alkaline by adding ammonia solution. After that, the volume of this solution is made up to 50 ml with the help of distilled water, and the entire preparation is kept aside for 5 minutes. Similarly, in case of standard solution, we use 2 ml of standard iron solution 20 ppm and dissolve the solution in distilled water and make up the volume of distilled water to 40 ml. Then again, similar to the test sample, here also we add 2 ml of 20% weight by volume of citric acid, and then 2 drops of thioglycolic acid are added to this solution, and the solution is made alkaline using ammonia solution. Then the volume of the preparation is made up to 50 ml using distilled water, and the entire preparation is again kept aside for 5 minutes. So after 5 minutes, the complex of ferrous thioglycolate is formed, which is pale pink to deep reddish in color, and the color of this complex is compared between the test and the standard solution. So continuing with the experiment, so you can see here we have taken thioglycolic acid, and this is the standard iron solution 20 ppm that we have prepared, and this is the ammonia solution that is required for the formation of the precipitate, and this is citric acid which is also required for the formation of the complex. And here we have marked 2 Nacl's cylinder as standard and test, and in the test we have taken 1 gram of sodium chloride and dissolved it in distilled water and made up to volume to 40 ml. And in case of standard, we have taken 2 ml of standard iron solution 20 ppm and dissolved it in distilled water and made up the volume up to 40 ml. So this has been done. Now we will be adding 2 ml of 20% weight by volume of citric acid. That means 2 ml from this solution, and we will be adding to both the Nacl's cylinder. Now we are adding 2 ml of citric acid 20% weight by volume here. To the test Nacl's tube, and here another 2 ml of 20% weight by volume of citric acid to the standard, and then we have to add 2 drops of thioglycolic acid to each of the Nacl's cylinder. we are adding 2 drops of thioglycolic acid to each of the Nacl cylinder. After addition of thioglycolic acid to both the Nacl cylinder, we are going to add few drops of ammonia solution to make the solution alkaline to both the Nacl cylinder. So
finally, as you can see, the color developed, the test sample, as you can see, is less intense. The color of test sample is less intense than the standard. So we say the test sample passes the limit test for iron. Thank you so much.
কি দেখবেন না বন্ধ করুন?